Hello, I'm Leslie Recio, the VP of Research and Development at ILS. Today I'll be talking to you about ILS capabilities with regard to safety assessments. ILS has been in business for more than 30 years with a number of commercial clients that span both pharmaceutical, ag chem industry, and food additives. We also conduct a number of client collaborative studies with academic institutions, including MIT. We have very strong scientific breadth. We include preclinical safety assessment and some efficacy studies, histology and pathology on-site, computational toxicology, and we do a number of client collaborative studies where we design studies to meet clients' needs. The scientific depth at ILS is very strong. 1/3 of our staff hold advanced degrees. We publish extensively at the request of our sponsors. We have both board-certified toxicologists and pathologists. Our histologists and animal care staff are certified in their special areas, and we are considered to be at the forefront of toxicology in that we participate in international meetings. We participate in the OECD process. For example, myself and my colleague Dave Allen have both participated in OECD guidance, regulations, and development of new guidances. We also participate in grant review panels, and we do a number of integrating of new state-of-the-art molecular biology technologies into traditional toxicology. The scientific focus at ILS is primarily safety assessment that includes in vitro toxicology, in vivo toxicology, genetic toxicology, histology pathology, molecular biology, and computational biology and information sciences. We are regulatory compliant with both FDA and EPA guidelines. Our study designs are designed to meet OECD guidance requirements, and we produce documents that are ECTD ready for the FDA. We have annual GLP training, SOPs, and many of our software packages we use to collect data are Part 11 compliant. We have a state-of-the-art animal facility that's been ALEC accredited since 1993, which has a fully equipped necropsy and surgical suite. The in vivo toxicology is compliant with all guidelines, ALEC accredited, as I mentioned earlier, and we conduct a number of traditional studies, acute, subchronic, chronic studies. This includes 28-day studies, 90-day studies, as well as in vitro micronucleus, in vivo micronucleus studies, and interlaboratory validations and mode of action studies. The animal species used at this facility include rats and mice, and the Provantis system that we use to collect our data is Part 11 compliant electronic data capture system. The histology and pathology is on site. We have a full screen necropsy facility as well as immunohistochemistry lab and histology lab. The immunohistochemistry includes stains for cell proliferation or cell death. We also have the capability of doing gene expression profiling from formalin-fixed paraffin-embedded tissues. The genetic and molecular toxicology group is compliant with ICH S2R1, also with EPA guidance requirements, and we do have flexible protocols that we can integrate multiple endpoints in the same study. We offer the full test battery, which includes the Ames test, in vitro micronucleus assay, in vitro chromosome aberration assay, both in CHO cells and peripheral blood lymphocytes, the in vivo micronucleus and comet assay, as well as the in vivo PIG-A gene mutation assay. We also conduct a number of experimental studies or follow-up studies using in vitro hepatocyte models such as HepRG, as well as gene expression profiling to get at mode of action and to understand what is going on in that biological system. We also offer a number of genetic toxicology screening assays. This includes structure-activity relationships using Case Ultra software. We also offer a mini Ames bacterial mutation assay and an in vitro micronucleus assay, high throughput screening assay that we can do with minimal amounts of compound. These screening assays are miniaturized versions of the definitive assay, so we anticipate they are highly predictive of the definitive assays. The gene expression genotyping is done in a GLP-compliant molecular biology facility. We do a number of assays, including quantitative RT-PCR for specific genes, as well as using state-of-the-art next-generation sequencing to understand gene expression profiling, which we use extensively for mode of action assessment. The information sciences group provides assistance in all stages of data acquisition management. They have expertise in toxicology, chemistry, and exposure. They also have a lot of expertise in preparing documents, dossiers on your compound, can do customized searches, and many of these individuals that do this work are Ph.D. level toxicologists, chemists, so they're able to not only provide you literature information, but also give you a peer review or a critical eye regarding what is the literature available to you, what are key studies, and put that all together in a single document. This also is the group that conducts computational toxicology, which includes structure-activity relationships, as well as conducting analysis of the Tox21 high-throughput data that's being used, part of the FDA, EPA alliance. Finally, ILS differentiates itself a number of ways from other CROs that we provide comprehensive, integrated investigative toxicology and toxicogenomic services where we integrate genomic technologies into traditional toxicology studies. We have very strong scientific knowledge, very strong scientific breadth, where we include toxicologists, pathologists, and molecular biologists, as well as individuals who are experts in alternative animal models. We also have responsive, experienced study directors that we ensure that they have a good client interaction. We do publish at the request of our sponsors. We integrate histology and pathology services in the same site, and we have been in business for more than 30 years with a very strong and loyal client base. Thank you very much for your time today.